Well, thanks everybody for your attention today. I'm going to be flying through a study we've been working on for the last 4 or 5 years. I've seen a lot of really interesting talks at this conference, so excited to share some work with you today. So we're interested in several research questions here, kind of how are trees responding to multiple simultaneous global change factors? They're not experiencing these things one at a time. And this is just a caricature of nitrogen deposition and ozone affecting a tree at the same time. We want to understand how these responses are similar or different among species, and if we can get functional trait information to try to start to delve into what explains some of that variation among species. And then finally, how do these aggregate to the stand level, and how do they affect the ecosystem services upon which society depends? So this is just a one-slide summary of some of the analysis. This is an analysis of about 1 million individual trees across the CONUS using the Forest Service FIA database. The original analysis represented about 90% of the forest biomass. We have since updated that to, I think it's about 95% now. The main responses of interest here are the growth and survival for about 150 tree species. We have 7 predictors that are all matched to the plot spatial location and the mean measurement between measurement intervals, or the mean value between measurement intervals. The 7 predictors are tree size, competition within the plot. I'll kind of dissect these later to give you more information on what these are. Temperature, precipitation, nitrogen deposition, sulfur deposition, and ozone. We use simulated annealing by species using 100,000 iterations and maximum likelihood, and assess multiple models with AIC. And we've also added an uncertainty assessment on a 5-point scale to this. The equation for growth is shown at the bottom, and I'll sort of decompose that in the next slide so you understand, why are we using this functional form? What does it mean? What do the terms represent? So here's the growth equation. I'm going to talk about these 5 sets of terms here. The first is an intercept term. If none of these factors affect the growth rate, there's just an intercept that's estimated. The size term is based on a power function. Larger trees tend to grow larger, which saturates at some point. So we use a power function, m to the z, where z is constrained to be less than 1 here, this blue curve. The next set of terms here is sort of represents fertility and competition. I won't go into a lot of detail on these, but it's basically to represent in more fertile environments. They tend to have higher basal area, or BA. The BAL term represents competition broadly, where if there's a lot of trees that are larger, that's what the L means, larger than the focal tree, then they may be outcompeted either for light or nutrients. The next 3 terms represent factors that we think can have positive and negative impacts on tree growth. So here we have temperature T, precipitation P, and nitrogen N. We use a pretty flexible 2-term Gaussian function that allows for flexibility of that relationship. And here I'm just showing you what those terms represent. So the X1 term, where here is T1, P1, or N1, controls where the location of that peak is. So here's just a caricature of changing it from 1 to 2. And if you estimate a very large value, you can approximate a line. The second term there controls the breadth of the relationship, where bigger numbers meet a wider curve. For the last 2 factors, sulfur and ozone here, we use a sigmoid function that's governed by 3 terms. And those 3 terms basically control the shape of that curve, just like before, where the S1 term controls the 50% reduction mark, the S2 term controls the steepness of the function at that 50% mark, and the S3 governs the floor. So what do we get? We do the simulated annealing for all these species, and what we get are these, I'm just going to show you one species to decompose it so you can get a feel for what these curves actually look like. This is an example for Abies balsamea, where I'm showing the growth rates, or the growth curves at the top, the survival curves in the middle, and then the histograms of the data at the bottom. And you can see that for this species, these are the 5 factors of interest: temperature, precipitation, nitrogen, sulfur, and ozone. And for this species, the growth rates tend to increase with temperature and the survival rates tend to decrease. And this is kind of a canonical response that we see with a lot of species. For precipitation, the growth and survival appears to peak somewhere in the middle. This salmon color is the range of data in the FIA. And so you can just interpret this is the estimated relationship to these factors. It also appears to be sensitive to sulfur, but not to ozone. So that's just an example. This is a screenshot of a folder on my hard drive. It just shows that there's a lot of files here and a lot of information. So we're talking about 150 species, 2 responses, and 5 drivers of interest. It's about 1,500 curves. So how do you describe that? How do you synthesize that? How do you get that information out? So we've decided to unpack this into 3 separate papers. The first I'm leading, it's the climate responses, where we'll focus on the temperature and precipitation responses that we just accepted last week in the Global Change Biology. The second is led by Forest Service collaborators here, Linda Pardo and others, that will focus on the deposition responses, nitrogen and sulfur. And the third will be led by the Park Service, co-led by the Park Service and EPA, that'll decompose the ozone responses. Today, though, with the focus on climate change, I'm going to just be talking about the temperature and precipitation responses. So here's all the curves. There's a 1 slide splash of all the relationships just to show you the richness of information here. The temperature responses are on the left, the precipitation responses are on the right, with growth and survival inside of those. And then they're grouped. The top row is whether it's an increase in relationship. The middle row is whether it's a decrease in relationship, and the bottom row is a unimodal relationship. So what can we do with these? What can we use these to try to understand? So there's lots of things you can do with these. You can use them to predict how species might respond to changes in temperature and precipitation. You can use it to compare the growth under an urban heat dome. You can use them to try to understand how forest trees are responding to temperature and precipitation. And here, T1 and P1 can approximate climate tipping points. This is where the response of that species fundamentally shifts from an increase in relationship to a decrease in relationship. So we're interested in lots of different questions to address with these curves. One is spatial variation in the response. And that spatial variation can occur at different scales. It can occur at these large regional scales, like is the Northeast responding differently than the Southeast? It can respond at very fine scales, like are all the trees in a plot responding similarly to local conditions? Or are there increasers and decreasers, or winners and losers, even at a very fine spatial scale? So here's a slide of some of those results. These are the effects at the CONUS. These are plot level responses to recent climate. This is 2018 and 2022 average conditions using PRISM. And then we are showing just the mean plot effect. So remember, within a plot, there might be anywhere from 1 to, I think the maximum is like 30 or 40 species. So there could be a wide range of responses even at the plot level. But here we're showing the mean effect under current conditions is the first column. Plus 1 degree centigrade are these two. You can see my cursor as I move it, right? Can somebody nod? Charlie, yes. I'm seeing. And a plus 2 degrees centigrade perturbation here. And precipitate, so temperatures at the top, precipitation's at the bottom, again, for growth and survival. And then for precipitation, our 2 scenarios were wetting by 25% and drying by 25%. And so you can see that on average, under current conditions, we're seeing an increase in growth rates throughout the mid-Atlantic relative to the mean conditions for those species, and a decrease in the upper Midwest and the far Northeast, parts of the Southeast, and the Rockies. And that as you get warmer conditions, you're essentially looking at where the dominant relationship at that point is for an increasing growth with small changes in temperature. And so areas that do well do even better, and areas that do poorly do even worse. And so that's why you're getting greener colors as you go to the right and browner colors as you go to the right. With precipitation, we see the opposite, that it's not necessarily trending in the same direction. As we know from IPCC estimates, that we do expect more wetting in the east and more drying in the west. And what we end up seeing is that's actually bad for both regions. So wetting tends to make it a more negative relationship in the east, and drying makes it a more negative relationship in the west. So this doesn't bode well for future forest responses. We can also use these to run what-if scenarios. So these are species level effects for incremental shifts in temperature. This is from plus 0.1 degrees centigrade to plus 4, and incremental shifts in precipitation from minus 80% to plus 120% for growth and survival. And here now we're looking at the median across species. This is not across space anymore. This is just counting each species once. The 10th percentile, this represents the more negatively responding species, or the less positively responding species. And then the 90th percentile represents the more positively responding species. So you can learn a lot from these what-if scenarios. It suggests that the median effects here are positive for growth, this blue color, so long as the window for precipitation stays relatively narrow, a minus 20 to plus 40% window. The median effects, though, for survival were negative across all scenarios we examined. Sensitive and tolerant species exist everywhere. So this is, the middle row looks red and the bottom row looks blue. That just means that there are sensitive and tolerant species everywhere. And we can also use this to look at what has been dubbed sort of these safe operating spaces for forests in the United States. So if we just say that 5% or less reduction in growth is pretty good for a forest, we can stay within that zone that's represented by this little white area here with up to a 1 degree shift, so long as precipitation doesn't change by more than plus or minus 10%. We can also stay in that zone for survival with up to a 2 degree shift if precipitation doesn't change by more than 10 or 15%, or 15 or 20%. And here now I'm overlaying the future predicted conditions under the RCP 4.5 and 8.5. And what you can see here is that even under the RCP 4.5, which is a solid box here, we're pushing a lot of these species into very negative territory. So we also did some cursory comparisons with experiments. 2 of them I'll show you here for Quercus rubra and Acer rubrum, compared with Reich et al. And Melillo et al. And so with the Minnesota experiment, in order to compare these curves to experiments, you need to understand both where the experiment was in terms of the conditions and what the perturbation was. So here that's represented by the blue circle here, where it was at 3.3 degrees centigrade and perturbed by 1.6. And since I'm running out of time, I will sort of speed to the end. The take-home message was that in general, these curves do match the few experiments that we actually have information for. But in some cases, that isn't the case. Agreements were not as good as survival, but a lot of that is because seedlings and trees are not necessarily responding similarly to global change. So we're using these in a lot of extensions and applications. The critical load, these are used to estimate critical loads for nitrogen, sulfur, and ozone, estimate changes in above ground carbon sequestration, timber and pulpwood. And we're interested in developing other potential applications for this work. There's a lot of things not included in these curves, and we're interested in sort of adding more factors or other things that affect tree growth and survival. We know that. CO2, pest, fire management. Many other, our climate factors are very rough. We know that things like vapor pressure deficit have a much more mechanistic link with tree growth and survival, but they're all correlated with these means because the trees are only measured every 5 years or so. So we can't really disentangle mechanism from a study like this. So the next steps are we're developing the recruitment response curves, revisit the suite of predictors to include pests, fires, and others, consider other statistical architectures, and partner with other groups like people here. So with that, I will stop and take any questions.
Great. Thanks, Chris. Really interesting work. Yeah, I'm glad. It's too bad we didn't have time for you to run through all 1,500 curves there for us. But there is one question in the chat. Maybe you can help respond to it there. But we do have to kind of move on here to our next speaker. Our next speaker here is Justin Coughlin. He is with the U.S. Forest Service Pacific Northwest Research Station.
Okay, great. Well, yeah, thanks everyone, and thanks for the invitation to talk today. Good afternoon, everyone. My name is Justin Coughlin. I'm an air quality specialist with the U.S. Forest Service, actually the Pacific Northwest region. I'm a biogeochemist by training. And it's fortuitous that I'm going right after Chris because I'm actually going to talk about how we're applying some of these curves, at least on the air pollution side, using a previous iteration of the curves, not the newly released, and I'll touch on that at the end. And I just want to mention that, a lot of this was done when I was still an employee at the U.S. Environmental Protection Agency. Kind of in the context, it was a research effort spun out of some policymaking efforts related to ambient air quality standards. So just hopping in, I'm going to kind of cover real quick just some air quality background and some of the trends that we've been seeing over the last 20 to 40 years. Nitrogen emissions have rapidly increased beyond pre-industrial times, primarily due to fertilizer production as well as fossil fuel combustion. And these emissions can eventually deposit back to ecosystems in the form of both dry and wet deposition. And while nitrogen serves as a critical nutrient for plants, it can also have cascading impacts that lead to unintended consequences in ecosystems such as soil acidification and coastal eutrophication. And we can model nitrogen deposition using emissions information, measurement data, as well as chemical transport models. Sulfur, back in the 1970s, roughly 60% of sulfur emissions were coming from EGUs, and this was leading to high sulfur deposition, notably in the northeastern US. This sulfur was directly contributing to soil and aquatic acidification through the production of hydronium ions, as well as base cation consumption. So you can see here from Driscoll et al., as a time series of sulfate ions in precipitation through time. And you can see the recovery that was occurring both in the precipitation as well as in the stream water as we started to reduce that sulfate deposition. And this really, started to come into public awareness through, the acid rain issues that were occurring due to red spruce and sugar maple dieback, in the 1950s through the 1980s. And this ultimately led to the Clean Air Act amendments of 1990, targeting these acid rain issues. Shortly after those Clean Air Act amendments were passed in 1990, you can see that we started to decrease the amount of sulfate deposition that was occurring as we were targeting those emissions. So just looking at some of these trends, here I'm showing sulfur dioxide emissions as well as oxides of nitrogen or NOx emissions through time. This is 2000 through 2020. And I'm also showing some of the US EPA regulations that were in place, such as the formerly applicable CARE, the cross-state air pollution rule, the mercury air toxics rule, as well as the SO2 data requirements rule. And these were really targeted at trying to reduce SOx and NOx emissions from power plants. Alternatively, ammonia emissions have stayed relatively flatlined through this period of time. It's not a regulated pollutant by US EPA. And so you can see here on the left, how these ammonia emissions over the last 20 years are relatively flatlined, as well as the concentrations that are associated with these. And these are nationally aggregated data. So, what's happened? So on the SO2 and the NO2 side, the ambient air concentrations of both SO2 and NO2, as these emissions have gone down, we've seen decreases in the ambient air concentrations of both of these. Similarly, we've also seen this trajectory of sulfur deposition coming down in tandem with the reduction of those SO2 emissions. However, like I've been saying, with this reduced nitrogen or ammonia or ammonium, we've seen this leveling off in total nitrogen deposition that's been occurring because we aren't really targeting those emission sources. So the next question, and Chris touched on this a little bit, is how do we evaluate this information against terrestrial ecosystems? Well, we use the framework of what is called a critical load, which is the amount of pollution that can lead to harmful changes in an ecosystem. These were originally developed kind of in the late 1980s and were developed using experimental studies. These are expressed in the terms of kilograms per hectare per year of nitrogen or sulfur, and that's in the form of deposition. So here I'm showing kind of two different response curves, one being monotonically decreasing, one being a unimodal response curve. And you can see where we set the critical load for these response curves, at the point at which we begin to see detrimental impacts. And over the last decade, there's been a plethora of critical loads research and development work that's been going on for different ecosystem components, such as trees, lichens, herbs, and aquatic acidification. And historically, these are evaluated critical loads in the form of an exceedance. It's binary. You're either above the critical load or you're below the critical load. And critical loads have been implemented or incorporated into a whole slew of policymaking efforts, such as flag guidance for federal land managers, US EPA's secondary national ambient air quality standards for NOx, SOx, PM, NEPA impact statements, as well as forest planning and wilderness stewardship efforts. However, with the development of these entire dose response curves, we're now able to kind of expand upon the traditional framework of exceedances and actually use the entirety of the dose response curve, as Chris was just showing for the climate side on the air pollution side. So this leads me to the research questions that I'm going to kind of run through today for this study that we had, which were, how have US forests responded to these declining air pollution levels? Are sensitive tree species being disproportionately impacted by atmospheric deposition? And are further atmospheric deposition reductions needed to protect forested areas? So as I mentioned before, we were using a previous iteration of these dose response curves from the Horn et al. 2018 publication, where they evaluated annualized growth rates and decadal survival probability and their relation to both nitrogen and sulfur deposition. In that study, they analyzed 94 different tree species that had at least a sample size of 2,000 trees within the national FIA data set. It's the same methodology, essentially, that Chris ran through, except the Horn et al. Publication did not include ozone, and it also did not have that sigmoidal function for sulfur. So we use these curves to evaluate how changing atmospheric deposition dynamics have either benefited or harmed species, and we mostly kind of honed in on the most sensitive species. So just looking at some of the data pipeline of kind of what we folded into this study, we used the national level FIA plot level data. We also pulled in continuous live tree basal area estimates from, if you caught any of the big map presentations earlier today, this was kind of a previous iteration, but was looking at basal area rather than above ground biomass. And so we use that as a live tree kind of proxy for species range, as well as the basal area itself. And here we're using a static forest cohort, so there weren't really any underlying dynamic vegetation models that were pinning or that went along with this. There's also an annual deposition data product that comes out of US EPA for both nitrogen and sulfur deposition. So we pulled in all 20 years of that data into this analysis. We also pulled in those response curves that I mentioned from the Horn et al. Publication. And what we ended up with were both plot level data as well as these continuous 250 meter pixel size raster data sets of individual tree species responses, and then also moved it into the basal area, framework. So I'm going to kind of focus here on like fifth percentiles and medians. And so, while I flip through this, when I talk about a species level fifth percentile effect, what I'm talking about is the most heavily impacted or worst impacted trees within a species range. There's the interspecific fifth percentile effect, which at a given point, what is the most, the worst impacted species within that, that pixel. And then at a plot level, we're looking at the, worst impacted trees within a given plot. So as I move through these fifth percentiles, that's kind of what I'm referencing. So when we look nationally, here we're looking at the median species effect for those 4 different endpoints. All of the slides I'm going to walk through, nitrogen will be on the left, sulfur will be on the right, growth will be on the top, and survival will be on the bottom. And here we're looking at the median species effect to both growth rate as well as survival probability. And we're looking at 2017 through 2019 average nitrogen and sulfur deposition. What we tend to see is that, for the most part, there are minimally negative effects occurring at the median level, somewhere between 0 to 5%, reductions in either growth rate or survival, with the exception of sulfur growth, and I'll talk about that a little bit later. And on the nitrogen side, you actually see increases that are occurring due to that nitrogen deposition, notably for growth. And so this kind of creates a paradigm shift for policymakers about pollution being exclusively harmful. And we have to kind of try to contextualize that. However, when we flip over to the fifth percentile effects or the more sensitive species, what we see are much more negative growth and survival rate responses across the CONUS, greater than 5% reductions in growth rate and survival. And even for nitrogen, you're seeing in certain hotspots in the northern forest, pockets of the west, and some of these agricultural regions, more harmful effects that are occurring. We also evaluated every individual species through time. So between 2000 through 2019 at all FIA plot locations. So here I'm showing plot level results. The green is the year 2000 and the blue is the year 2019. And so we are looking through time, how are individual tree species responding? So this is the fifth percentile for that given species. So again, the worst impacted trees within that species range. And what we see are, many species experience positive increases in growth as pollution has decreased, notably for sulfur. So you see this movement towards kind of a 0% growth rate reduction or baseline through time. Oop. But as I've been mentioning, not all species are responding positively. Some species that are nitrophilic have actually seen reduced growth as we've been decreasing nitrogen throughout the last 20 years. So when we pull all this together, here I'm showing the interspecific effect. So this is all 94 species through time. The brown is showing the deposition trends nationally across all states. The blue is showing those fifth percentile interspecific effects, and then the green are the median effects. And we're seeing that these regulations, are doing what they're supposed to be doing, and we're seeing, shifts towards more positive growth, through time as this deposition has come down. Once again, that nitrogen deposition caveat, because we don't regulate reduced nitrogen, we've seen a flatlining in the mid 2000s that hasn't quite been addressed at this point. So this begs the question of how much further do we need to go? So we can kind of flip the question on its head. So instead of looking at the effects, growth rate reduction, we can also ask how much detrimental effect do we want to prevent? So here we chose 5% reductions in growth and 1% reductions in survival as thresholds that we want to protect against. And we selected 95% of species as the threshold that we want to protect within forests. So what we see here are that reductions of nitrogen and sulfur are needed across much of the eastern US, as well as pockets of the western US. For sulfur, there isn't much reduction needed for in the west because sulfur is already pretty low. Again, these agricultural hotspots tend to jump out, notably on nitrogen survival, places in the Midwest, as well as in North Carolina. The last result I'll talk about here is we folded all those forest health indicators into one figure to look at the total basal area impact for 2017 through 2019, average nitrogen and sulfur deposition. So here, we're actually using actual basal area estimates to look at where we're seeing basal area increases due to nitrogen and sulfur and where we're seeing decreases. So in the eastern US, we're seeing predictions for large increases in basal area due to the beneficial effect of nitrogen on growth. Whereas in the Western US, we see these largely negative impacts. And we think that a lot of that is due to these really low sulfur critical loads that came out in the Horn et al. Analysis because of the functional form was monotonically decreasing. And I see you, Sean. I'll wrap it up here in a sec. So with this new analysis that Chris just ran through, like I said, we had monotonically decreasing responses in the Horn et al. Publication for sulfur. Now with this tabletop function or sigmoidal function, as you can see here for Douglas fir, we expect to have higher confidence in our results because the sulfur sensitivity won't be so low. Just looking at kind of a preliminary look at it here on the x-axis, we're looking at the new analysis that Chris was just presenting and the Horn et al. Analysis. And what we see are a shift towards higher sulfur critical loads in the new analysis with that new functional form. So now we need to reprocess that entire spatio-temporal data set with the new results. And these data can assist policymakers in establishing future national ambient air quality standards. The last piece I'll just mention, just kind of communications and outreach. The Northern Research Station has recently released a tool called the Critical Loads Assessment by Site tool or CLASS tool that is going to include all this information, both the critical loads as well as the percent effects to both growth and survival probability. The Washington office for the Forest Service has also been releasing the Air Pollution Effects on Forest Handbook, Volume 1 Trees, details all the Horn et al. Analysis. And then lastly, if you want to find out more about critical loads, there's a recently released Critical Loads Hub, which goes through all the background as well as how you can use critical loads within resource management. So I'd just like to acknowledge these folks. And if you have any questions, I assume I'm over time, so you can just direct them in the chat. And again, appreciate the opportunity to speak today. Thanks so much.
Great. Thanks, Justin. Really informative. Appreciate it. Okay, moving right along here, we have Charlie Canham. He's with the Carey Institute of Ecosystem Studies.
Great, thanks. Many of you know that I've been working with FIA data for many years. And about 10 years ago, Laura Murphy and I did analyses very similar to what Chris described, in which we simply used conditions during the 5-year census intervals to look at growth and survival. We tend to be working with the 50 most common species in the eastern US because the information is being used to inform the modeling we do. And, we did these papers and we published them. One of the key results was, and a little surprising to me, was that frankly, neither growth nor survival responses to climate seem to map those species onto their existing ranges. So using a 5-year average temperature or precipitation measurement during the census interval produced responses that had little obvious relationship to the actual geographic and temperature precipitation gradient distributions of these species. And even more surprising, in about half of the 50 species, there was actually no variation in survival as a function of temperature or precipitation across the eastern US when using those 5-year average climate variables. I'm sure we're all comfortable sitting at home or sitting in our offices doing this on screens, but I actually miss being at the physical meetings. I guess it was the meeting in Baltimore, the stakeholder science meeting in Baltimore in 2012. I happened to sit down next to Rachel Ryman, got talking, and mentioned that I really wished I could get my hands on the tree ring data that FIA folks analyzed back in the 1980s to look at air pollution effects, which turned out to be a very difficult thing to do with the tree ring data, and very little came of that. But Rachel said, oh, I have some CDs that are labeled tree ring data that were left behind when someone retired. It took us about 2 years to reconstruct what was on those CDs. There were basically the data from 23,000 tree cores collected by FIA in the 1980s. And I will freely admit that I had to, so I thought, well, this is really exciting. I'm going to see what I can learn from this. And to be honest, I decided to try something new simply so that I could get this published because people have been looking at climate responses using tree ring data for decades. And so that figure in the upper left-hand corner A is, imagine a whole series of cores collected across a very broad geographic range. And you'd be interested in trying to fit that dark line would be what you'd be trying to fit the temperature response, for instance. And any individual core would be cover just a portion of that range. And you'd essentially assume that the individual core data just varied randomly around that line. But B is an alternative in which individual cores would show patterns that were very different from what the broader geographic trend was when temperature was at the long-term mean. And so a number of us, with Rachel, Richard McCulloch, and Liz Burrell, and Laura Murphy, and I published a paper 6 years ago now in Ecosphere in which we used the tree ring data to look for these interannual, whether the long-term responsive growth to long-term mean temperature differed from the response to interannual variation around that mean. And so basically the model we're trying to fit is characterized in panel D, where the dashed black line is what you'd expect when temperature is at the long-term mean at an individual location. But within that core at that location, interannual variation would be reflected in the shapes of the red lines. So, we only had, there were 14 species represented in that tree ring data set, all sampled in the Northeast, sort of Ohio up to Maine. But as you all know, with 20 years now of annualized surveys, we can essentially do the same thing using imputation to try to fit essentially identical models using the FIA census data. And I'm not going to try to describe the functional form. It's about 60 lines of our code. But basically, given that the census intervals overlap, you can impute the effects of annual variation in climate exactly the same way that we did with the tree ring data. So we've tested 3 alternate models. The first is whether there's any climate effect at all. The second is whether the only response we see is sort of that black dashed line. And the third is whether we see whether AIC supports a model in which there is a different, the red lines representing the local response to interannual variation around the long-term mean. Okay, the data sets, we, again, have been working with these, the 50 most common tree species. Sample sizes are range from a small of just under 10,000 to over 350,000 for red maple. My dog thinks it's dinner time and is sitting behind me whining. So we look at both saplings and adult trees, and the most parsimonious models do in fact fit separate parameters for saplings and adults. We fit the model simultaneously with a common size function. We use PRISM data at an 800 meter resolution and true plot locations under an MTA. And then we use the period from 1960 to 1999 as our baseline to define the long-term mean climate. Here are the just a quick summary of the results. As with Chris, I don't have nearly as many figures to look at, but way too many to go through. For 41 of the 50 species, models that do include this local adaptation acclimation are far superior to models based on absolute climate. I actually think it's probably just data availability that we don't see it in the other 9 species because I can't see any obvious correlates. And the models explain a reasonable fraction of the variation in absolute growth. So here's an example with saplings of red spruce. The temperatures on the left, precipitation on the right. And it's important to note that tree growth, tree dendrochronologists have been for years fitting models that look at both current and previous year effects, given the nature of carbohydrate economy in trees and the importance of carbohydrate stores from the previous year for growth the next year. In red spruce saplings, the main effect of temperature is in the current year and very little effect of previous year. But very little variation in red spruce sapling growth across a temperature gradient when temperature is at the long-term mean. On the other hand, a fairly tightly adapted local response to temperature, with saplings doing better when temperature is slightly cooler than the long-term mean at a location. The pattern for precipitation is somewhat similar in that current year precipitation matters more than previous year. The long-term trend, or the geographic trend, is for growth to decline with increasing precipitation, but at any given location, when it's wetter than average, growth is higher. So that's what those, so here are just a couple more examples for red maple and paper birch. And this is a fairly common trend in which, by and large, trees grow better when growth in either the current or previous year is cooler than the long-term mean, but they grow better when the precipitation is higher than the long-term mean. This is a perhaps horrifying figure, but after trying to stare at all those figures, I finally decided that I needed some way to graphically sort of synthesize this. And so what I'd like you to look at for precipitation and temperature are the blue and red terms here. These are illustrating how much variation in growth we could predict from the models as a function of variation in one of the terms in the model when everyone else was held at their median value. And so if the dark blue is broader or wider than the light blue, then the regional mean is more important than local variation, and vice versa. The same with red for temperature. By and large, the darker bars tend to be, which are the regional effects, tend to be larger than the interannual variability effects, but there are lots of exceptions to that. So red oak, for instance, has a very strong response to interannual variation at the local, relative to the local long-term mean, a much smaller response across the geographic range of the species. The same would be true for Liriodendron, with local precipitation having a much greater impact. One thing to point out is that I suspect you all know, that compared to our baseline period, interannual variability during the past 20 years has increased for precipitation and decreased for temperature. So here's the real question. Is this genetic adaptation or phenotypic acclimation? Our results suggest, and this is very similar to the results in the 2018 paper with the tree ring data, that trees tend to develop an architecture and physiology that maximizes growth in years with higher than average long-term temperature, higher than average long-term soil moisture availability, i.e. cooler and wetter than the long-term average. But we have no real idea whether this is genetic adaptation or phenotypic acclimation or both. I've argued in the 2018 paper that parsimony suggests phenotypic acclimation because I can see how trees could develop this through, for instance, changing root shoot allocation, for instance. On the other hand, the fact that even saplings show very clear evidence of this local differentiation would weigh towards arguing that there's a genetic basis, or that these plants are able to develop the phenotypic acclimation very rapidly. And fundamentally, this is probably obvious, but the implications for responses to climate change are just fundamentally different. If the patterns are genetic, these trees adapted to a disappearing climate, or if it's phenotypic, are they capable of very rapid phenotypic acclimation? So just to wrap up, I suspect many of you know that much of my work with the FIA data has been designed to inform and develop behaviors for our models, or TND, spatially explicit individual-based tree model that I've been working with for over 30 years now. When we first ran this model with the original 5-year mean data, we saw very little in the way of climate change impacts. It took several hundred years because the strongest impact we found was on seedling recruitment. And so the real question is, with these new analyses, will the story change? So Sean, I will stop there and see if there's any time for a question or two.
Great, awesome. Thanks, Charlie. That's really really neat to see sort of the effect of local and regional effects on growth here. I think that's a really important twist on the stories here. There is a couple questions in the chat, but maybe I'll just let you respond to those in the chat so we can kind of just keep moving along here. But yeah, I really appreciate your time today. Thank you.
Sounds good.
Our next speaker is Nick Dewez, who joins us from the University of Toronto. Nick, can you share your screen and we'll get started?
All right, wonderful. Thanks, Sean. And thank you all for joining my talk about simulating future drought impacts on above ground biomass in East Texas under climate change from my work at Texas A&M University, Kingsville. Before we get started, I'd like to extend a special thanks to my co-authors listed here on screen. Forests play a pivotal role in regulating the Earth's climate and supporting biodiversity, extending a myriad of ecosystem services to human society throughout history. Ecosystem services that are becoming increasingly threatened by the rate and amplitude of projected increases in temperature and changes to precipitation regimes. Forest disturbance dynamics, also sensitive to climate, have profound impacts on nearly all aspects of successional dynamics and affect ecosystem services both directly and indirectly. Disturbances alter ecosystem and successional states, and future recovery patterns may differ from historical norms as a result of climate change-induced shifts in the growing environment. Late successional environments may seem unresponsive to change, while the underlying regeneration niche may be shifting. Before diving into the research, I want to quickly touch on simulation modeling as a whole. Major types include growth and yield, succession, and process-based simulation models. And it's also important to highlight the principle of parsimony here, which by ensuring model simplicity while also capturing adequate representation of the processes being studied. By choosing the least number of variables, processes, and spatial and temporal resolutions necessary to simulate the landscape patterns and processes relevant to the research question. When used effectively, simulation models are powerful at evaluating questions relating to forest change, disturbance, climate change, and management strategies in forests over large spatial and temporal scales. The 2011 drought in Texas was the worst in recorded state history. It killed approximately 300 million trees across the state and approximately 65 million in the East Texas region. The East Texas region saw increased mortality for several years following the drought, which was primarily attributed to insect and disease, while trees were left in a weakened state. Since then, some studies have shown that forests within the region were found to be resilient or resistant to the drought impacts, with spatial and species-based variations in these responses. The frequency, duration, intensity, and severity of droughts have significant implications for forest ecosystems, and climate change is expected to have an impact on these variables. The accurate assessment of disturbance impacts is crucial for future forest management and for policymaking. The increasing frequency and severity of disturbances introduces uncertainties about how forest dynamics will respond to them in the future, and the effects of drought as a recurring form of disturbance have yet to be simulated in East Texas. Which leads us to my following research question, which is, how does climate change affect the relationship between drought and forest biomass in East Texas? To address this, our study takes place in the central area of the East Texas region, and more specifically in the 4 national forests of East Texas and their surrounding areas. The figure on the right here illustrates the distribution of the 4 national forests within the East Texas region and the 10 land type associations that will be used in the simulation modeling framework. We omitted one of these land types from our simulation results as it is primarily composed of the water bodies within our study area. Approximately 57% of the East Texas region is forested, and historical disturbances include approximately 24 wildfires per year, hurricanes making landfall 2 out of every 3 years, and 5 major droughts have been reported over the course of the last century. Data for these climate change scenarios was obtained from PRISM 96 at Oregon State and from the Coupled Model Intercomparison Project Phase 5, which was available for the 80 years of the study. Temperature and precipitation data were only available to the current year, however. Therefore, we used a 30-year moving monthly temperature and precipitation average to replicate data from January 1991 to December 2020 through 2099. For the low emission and high emission climate change scenarios, we used historical data up to 2020 to provide each simulation scenario with the most realistic spin-up period as possible. To model landscape level disturbances and processes, the 80-year study period, we used the Landis 2 model. This is a spatially explicit stochastic model of landscape level forest dynamics used to simulate forest succession and disturbances and project changes in biomass and species distributions over a given study area. Spatially explicit model is used to answer scale-related questions in population ecology, and the stochasticity component refers to the property of being well described by random probability distributions. In addition to the base software, we used an additional 2 extensions, the biomass succession extension and the base drought extension. We also used PNet2 for Landis 2, which is a program that is a process-based model for carbon and water dynamics in forest ecosystems. We use this program to calculate SEP and ANPP, which are 2 important input parameters for the Landis 2 biomass succession extension. Landis 2 input data can be categorized into 3 groups: species data, land type association maps, and initial community maps. For our species inputs, we used FIA data comprised of the 14 most relevant species reported in the FIA database from 2000 to 2020 in the 4 national forests of East Texas. You will see the species themselves and some of their basic attributes in the table on your screen. And the land type inputs for Landis 2 are defined by the ecological processes under investigation, and heterogeneous landscapes are classified into homogeneous land types based on similarities between topography, soil types, and climate regimes. I won't go into any additional depth, as we've already discussed these when looking at the study area. The final major input for the Landis 2 is the initial communities. We use 10-meter land classification maps of central East Texas, sourced from the Texas Parks and Wildlife Department. These maps are based on detailed vegetative classification from fixed ground sample points across the state, and each color you see here on the map represents one of the species within each ecoregion. Within each pixel, age species cohorts are defined, and species age inputs were calculated with FIA data discussed on the previous slide. The input parameters discussed previously were grouped into 9 unique simulation scenarios seen here and replicated to account for the variability in the random number generators of the program. However, variation in above ground biomass outputs was found to be marginal within scenarios. Therefore, we limited this to 3 replicates per scenario. At the top of the table, you can see the historical projection scenarios that include our control climate data. With these scenarios, we simulated the relevant species of the region from 2019 to 2099, and the SEP and ANPP outputs of the PNet2 program were used as input values for all 9 scenarios. We evaluated how biomass differed between the 9 scenarios within 9 of the land types modeled. Land type 1 was excluded from this analysis because it was primarily composed of the water bodies within our study area. We used a type 3 ANOVA to evaluate the significance of the main effects and the interaction between climate scenario and land type. We then extracted marginal means and ran pairwise comparisons to evaluate the effects of climate and disturbance scenario combinations within our study area. On this slide, you can see a map on the left showing the initial communities for the historical projection climate scenario. And on the right, you'll see a simulation of the above ground biomass under the exceptional drought scenario, coupled with the historical projection climate data. Above ground biomass shows an increasing trend from 2019 to 2099 amongst all climates and disturbance scenarios, with the highest rise seen in the high emission, no disturbance scenario, and the lowest in the low emission, exceptional drought scenario. We found that overall biomass increases throughout the study period under all simulation scenarios. We also saw within a couple of the low and high emission scenarios an initial drop in above ground biomass, followed by recovery over the remainder of the study period. Other studies using Landis-2 have also seen this and found that the initial decline in biomass within climate change scenarios was attributed to climax species suffering heavy losses in the early stages of climate change simulations. Our pairwise comparison yielded some interesting results. As expected, a significant difference in above ground biomass was observed between most scenarios. However, we did not see a significant difference in above ground biomass between drought and no drought scenarios within the same climate scenario, except for the high emission scenario where this was significant. One of the major questions researchers in the East Texas region have been asking is whether oak or pine species are more susceptible to a changing climate. Therefore, we chose to analyze our species results by two groups, oaks and pines. And in general, all species saw a decline in above ground biomass over the study period, were oaks, and all pine species saw an increase in above ground biomass, except for shortleaf pine, which showed a variable response. When looking at the results of our pairwise comparisons, we found some interesting results when it comes to the above ground biomass of the species tested. Mainly that within a given climate scenario, pines did not show a significant difference in above ground biomass between any of the disturbance scenarios, whereas oaks only showed no differences between the drought and no drought scenario within a given climate scenario, similar to the overall above ground biomass results seen earlier. In conclusion, we found that our historical projection scenarios held the highest AGB values over the study period, with the low emission scenarios coming in second and the high emission climate change scenarios coming in third. Above ground biomass grew under all scenarios, varying between 0.1 and 8.9%. And we found that pines are likely more robust towards climate change than oaks because pines saw no difference within a single climate scenario, regardless of the disturbance involved. And oaks saw no difference between drought and no drought scenarios only. Moving forward, it's important to investigate the policy and industry implications of drought and climate change in East Texas. Investigating future drought impacts across the other regions of Texas is also an important factor. And using long-term data such as FIA for understanding the long-term forest dynamics in the region is critical. With this, I'd like to make a few acknowledgements. This work was made possible by the USDA Forest Service FIA program, the U.S. Forest Service Southern Research Station, and the Texas A&M Forest Service, and the National Forests and Grasslands in Texas. I'd also like to extend a thanks to all my co-authors again for their contributions and support with the project. And with that, thank you for your time, and I'd be happy to take any questions either in the chat or in person here, depending on the time available.
Great, thanks, Nick. Really appreciate that. Looks like we have time for maybe one quick question if somebody has one. Otherwise, we'll have to move on.
Just wondering, one thing I always think of with drought is fire, and I'm wondering, does your data, with your historic data, incorporate the losses that might have related to both drought and fire, or would one want to add another component of the model looking at biomass that looked at, like, could fire behavior change under these scenarios?
Yeah, so in this case, we isolated drought as a component moving forward, but in some of the historical work that we did do in the region, which wasn't discussed here, yeah, that there is a relationship with fire seen there. But here we were just aiming at isolating drought as a recurring form of disturbance through the end of the century.
Thanks again, Nick. Appreciate it. Let's
Thank you.
move on to our next speaker here. Our next speaker is Jeremy Groom with Groom Analytic.
Perfect. So today I'll be talking about some explorations into looking for evidence of tree migration in the west coast of the US. And I want to extend a special thanks to Oregon Department of Forestry, who provided a great deal of the funding, and also my co-author, Vicente Monleon. So I have in case you don't want to see a talk, I've got a dashboard at the tiny URL FIA tree mig site, and you can explore some climate things or maps of species distributions and what happened there. And also a link here to my paper. Came out in 2023. So background, climate change is associated with some geographic range shifts of trees and other taxa. And certainly, I think we're well positioned in the US with FIA data because it's extensive and rigorous. And the design allows for design-based estimates with revisit data, which I think is pretty cool. So how do we know if a changing climate is affecting distribution? Along the East Coast, there's been some work in the last numbers of years on looking at distributional changes in latitude and elsewhere, and looking at elevation. And with some assumptions of changes in latitude or elevation correspond to a response to specific predictors, climatic predictors. In 2015, Vicente and Lintz, they have a paper in which they used PRISM data to find mean FIA plot temperatures for those plots that had saplings and those that had adult trees. And they did this for all the plots in California, Oregon, and Washington. And then asked, okay, are the average temperatures of the plots for seedlings or saplings or trees different, and how different? So they were trying to use that as a metric of migration. So what was nice about this is it sidestepped assuming an elevation and latitudinal relationship with climate variables. Certainly, parts of the West, you could be going up in elevation and heading southward at the same time. So they were constrained by having a single visit data set at that point. And that's where this study came in. And so we're looking at evidence of tree species distribution shifts for those states. And it's largely the same data set, except we now have revisit data. And we're also looking at PRISM values for precipitation, temperature, and then maximum and minimum vapor pressure deficit. And we looked at two different metrics, one for range shift, that is, we're looking at the difference in mean climate variable values of occupied plots. So which ones winked on or winked off or stayed occupied. And density shifts were differences in mean climate variable values between plots. So which plots gained in trees and which plots lost trees, and what was the difference there? Just to run through this methodology, because I find it something of a mind bender. So you find the mean of occupied plot climate variable, like the temperature, for value for each species. So if we're looking at a single species here, in that box, we've got some yellow squares, and those will be FIA plots that have our species of interest. And right in the coloration is the first visit temperature gradient. So the cooler plots are on the left and the warmer plots are on the right. Given that, we can ask what's the mean temperature of those occupied plots. In the second visit, we're going to use the same temperature gradient. If we switch to the second visit's temperature gradient, if the trees were great at getting up and moving, then you might not see any change at all. So that's why we stuck with the first visit gradient. So what we're interested in is the difference in the two means between visits. So if you take the second visit minus the first visit and it's negative, then across the temperature gradient, the range of species X is shifted towards a previously cooler area. So that was range shift. For density shift, somewhat similar. We're going to find the mean of climate variable values for plots in which the species numbers increased and decreased. So here's that gradient again. And here's a plot that gained trees of a particular species, and the purples are plots that lost some species, some individuals. So we find a mean for both. There's the one that gained, lost, and again, find their difference. And so we ask, so what happened with the temperature change for those with more trees minus those with fewer trees is negative. So across the temperature gradient, the density shifted towards previously cooler areas. So there's an inherent assumption in this procedure. It is the same procedure that was used by Monleon and Lintz, but we're only considering the initial visit climate value for this analysis. And so we did have a question of, did climate values for plots change between visits and how? You could have an increase in all plot temperatures by a certain amount, although that's probably unlikely. And then it might not be even across the range. Perhaps plots that had higher initial temperatures would increase more. So this is a univariate design-based analysis, and it'd be really nice to include both what the initial value was and what the change was. And I gave that presentation 2 days ago on how we're trying to parse that. So as far as the climate variables go, we just poked at this with this paper, asking, well, what did happen in these plots? And this is on the dashboard as well. So all those points right there are FIA plots with, on the x-axis is the initial value, and then the change, the y-axis is the change. And then just very, very simple model selection of, hey, did an intercept term or a linear term fit better? And then track which species and how many species were better fit by each and what sort of a slope there was. So for the analysis, we relied on ratio estimators, well, difference in ratio estimators. So for the ratio estimators, got them from the green book. And then for the variance, we relied on Wolter. And a gnarly variance equation. Okay, so results. What we've got here on we have lines at 0 for temperature, precipitation, and maximum and minimum vapor pressure deficit. And then where the tree species estimates lay and whether or not their 95% confidence intervals overlap with 0. If they didn't, the dots in the middle were black. So what I see is that there really weren't many hits. So except possibly for minimum vapor pressure deficit. And in fact, yes, you would expect these sorts of results by chance. We did, for minimum vapor pressure deficit, ran a permutation test and determined that that one's unlikely to have occurred by chance. So maybe there's a shift towards plots with initially low minimum vapor pressure deficit. I mean, not all, there's one plot that shifted to the right, although the other 6 shifted to the left. I'm not going to look into it that far. When we looked at density, we're now down to 30 species. The story was a bit different. So we had quite a few more hits. And there's gray bars there. The gray bars were actually present in the previous figure, but they're very small. What they are is 95% confidence intervals for a generalized least squares estimates for what the estimate is for all of the species by a particular climate variable. And so there was a overall shift towards cooler and previously drier plots for these species. And certainly the numbers of species, they did, well, for minimum and maximum vapor pressure deficit, things are a little bit more all over the board. There was not a general shift in that generalized least squares result. However, numbers of species appear to have changed their densities. As far as the climate variable went, and these are from the dashboard, these figures, it looked like temperature maximum and minimum vapor pressure deficit generally increased, and precipitation generally decreased. For temperature, 25 species were better modeled with negative slopes, that is, their plots. And by visual inspection, it just appears that temperature increases were greater at initially cooler plots. And for precipitation, 26 species were better modeled with negative slopes as well. And precipitation declined most in areas with more initial precipitation. So we really didn't see a strong signal of range shifts. And maybe there's something for minimum vapor pressure deficit. And this does align with findings from elsewhere. In this case, why didn't we see results? I think there could be a number of reasons. So statistical power, we're trying to look at changes in plot occupancy in a 10-year period. So it could be just we didn't have much of a shot. There's also it could be ecological. It could be difficult to shift one's range into other species ranges due to competition, lack of seed bank, and so on. Also, the metrics themselves may be indirect. That is, temperature and precipitation might not be the best tools to use to try to gauge where species ought to go. And finally, and I think this ties in well with what Charles Canham was presenting on, but at a different scale, some of these species are spanning quite a big geographic area. And so we could be running into Simpson's paradox where they're not seeing patterns is kind of masking the fact that at smaller scales, there really are patterns that are present. We did see density shifts towards wetter and drier plots. And that's a little concerning because if that is so, then this could be evidence that there are contractions in species ranges. That is, they aren't maybe shifting their ranges, but they could be shifting within their ranges. So these findings are extremely general, and our hope is that the results assist others with their deeper exploration of individual species. And I'd be happy to take any questions.
Great. Yeah, thanks, Jeremy. That's really interesting work. We do have a couple of minutes for questions if anybody has a question for Jeremy.
I have a question.
Yes.
Sure.
Hey, awesome analysis. I was just wondering, I may have missed it, but were you using adult data only for those range and density shifts, or were you using the sapling and the seedling data as well?
We were just using, anything greater than 5 inches DBA.
Okay, cool, interesting. Thank you.
Yeah.
Thanks again, Jeremy. If there's no other questions, we will move on to our final speaker for the day, or for the session, I guess. Our last speaker for the session is Katie Baer. She is with the U.S. Forest Service and the Pacific Northwest Research Station.
Great. Well, thank you for the introduction, Sean, and thanks to everybody for sticking around for this last talk in the session. I'm going to take us from the overstory into the understory. So I am the shrub contingent of this trees and shrubs session. And today I'm going to be talking to you a little bit about a project I've been working on, trying to understand how climate change might shape the distribution and abundance of a couple of really important subsistence species in the forest of Southeast Alaska. So let's see, oh, there we go. So just to orient us to where we're talking about when we talk about Southeast Alaska, that is the area shown here in this green box. And more specifically, today I'll be talking about forested areas within the Tongass National Forest. So those are the green areas inside of the green box. Within Southeast Alaska, a lot of the communities, most of the communities are pretty isolated, pretty difficult to access. They're not connected to a larger road system. So the only way in or out is by boat or by plane. And what this can mean in effect is that food is very expensive in these communities. And a lot of the food that comes into these communities are low nutrition, shelf-stable items that many people supplement by going out and harvesting in the understory of the Tongass National Forest. Many of the communities border the Tongass. So not only are these species that folks are harvesting from the forest important from a food security standpoint, but many of these communities are either predominantly or largely Alaska Native. So these species that are being harvested from the forest also have a really significant cultural value to people that harvest them. And two of the species that I'm going to be talking about today are the two most common, I guess I should say that's three species, but they're the three most commonly harvested species in the forests of Southeast Alaska. That's blueberry right here, Vaccinium ovallifolium and Vaccinium alaskense. They're so phenotypically similar that they kind of get lumped together. And then salmonberry, Rubus spectabilis down here. And I've had the good fortune to be able to go to several communities in Southeast Alaska over the last few years and have conversations with people about what are your concerns for these understory species. And one that gets repeated over and over and over again is, I have seen the climate change within my lifetime. I know more climate change is coming to this region. What is this going to mean for these plants that I rely upon, both for food security and as a really important component of my culture? So with that, the objective of this project was pretty much twofold. First, answering that question of how do we expect climate change to impact these understory shrubs? And then, are there environmental attributes associated with greater abundance of those shrubs that maybe can help guide managers if their goal is to either ensure continued access or maybe promote the performance or abundance of these species moving forward under future climate change scenarios? So to get at this first question of what are the potential impacts of climate change, what I did was built an ensemble species distribution model using this amazing phase 2 understory vegetation data set that FIA collects. And I used data on occurrences of these species all throughout their Western US distribution, so all the way from Montana to coastal Alaska, and paired those with topographic data and then climate data taken from Climate NA for the 1990 to 2020 normals period. I then built projections under climate normals for what we think the distribution of these species will look like under normal conditions, and then projected that forward under 3 future climate change scenarios, shared socioeconomic pathway 2, 3, and 5 for 3 years going towards the end of the century to see what kind of trajectory we see. So 2050, 2075, and 2100. And with this, I was trying to answer kind of 2 related questions. The first being, what kind of changes do we see in binary predictions for occupancy of these species? And then how does the more continuous predictor of climatic and topographic suitability change under these climate change scenarios? So just to orient us to these maps, because we're going to be seeing a few of these in the coming slides, here we have maps of Southeast Alaska with the forested areas of the Tongass, colored in either blue or pink. Blue indicates an area that's projected to be suitable for the occurrence of these species, and pink indicates an area that's climatically and topographically predicted to be not suitable. So this is kind of taking a climate suitability prediction and just binarizing it. Basically, all I want you to take from this slide is when we look at blueberries, when we look at salmonberries, Southeast Alaska is a good place to be a blueberry and a salmonberry, climatically and topographically. Over 99% of the area is suitable for the occurrence of these species, minus a little bit of area up here near Haines and Skagway for salmonberries. And these statistics just tell you the models were a good fit. There's a pretty high level of confidence in those model predictions. So taking that, then we can compare it to predictions for future climate scenarios. This is just one. This is SSP3 for the year 2100. But this pattern kind of holds to some degree or another for all of the future climate scenarios in all of the years. What we're seeing is occupancy is expected to remain pretty high. So that's a really good climate change story. We're not going to lose these species in Southeast Alaska. But what is a little bit concerning is that there are areas, mostly inland, where we're expecting to see either, we'll say decreases in occupancy, losses of these species, at least, in smaller areas. And some of these areas are occurring in the vicinity of communities where these species are harvested currently. So near Skagway, near Haines, in the case of blueberry, near Hyder. So Across the board, we're expecting net losses in occupancy, but we're never dipping below about 95% of the area being suitable for the occupancy of these species, which is good. Now, if we look at, rather than binary predictions for occupancy, we look at this continuous variable of climatic suitability and kind of overlay climate normals versus future climate projections, this is what we see, which is a little bit more concerning. So areas here shown in red are areas where we're expecting climatic and topographic suitability to decrease under this future scenario. Areas in blue are areas where it will increase, and then sort of the degree to which, so deeper reds indicate a greater decrease, deeper blues a greater increase. And what this is showing is across the board for blueberry, climatic suitability is decreasing. So we're not necessarily going to lose it, but the suitability is potentially nearing that tipping point. It's a little bit more complicated for salmonberry. We do see predicted losses in climatic and topographic suitability the more inland you go, but out in these outlying islands, things are actually supposed to be improving. So that's kind of an interesting result. And one thing that I started thinking about looking at these maps is there's a body of literature. The results are a little bit equivocal, but in some cases, suitability for occurrence can be an indicator of the performance of a species. Sometimes it differs according to which aspect of performance we're talking about. But I thought, well, let's go ahead and run the analysis and see how predictive is suitability for something like cover, which FIA records on their plots for these species. And what we see is there's a very strong positive relationship for blueberry among suitability, our x-axis here, and percent blueberry cover. So what could be inferred if this relationship is maintained moving forward under climate change is here's our mean suitability under historical normals. And as we can see, this pattern holds across the different climate scenarios. We are marching down this curve to the tune of, by the time we get to 2100, this would predict, if this is maintained, a decline in average cover of the species of about 9.8 to 13.8%. So it may still be there, occupancy may remain unchanged, but the abundance could be significantly lower than it is currently, and that is concerning. On the plus side, no such relationship seems to occur for salmonberry, at least not for plots falling within the Tongass. If I do this analysis nationally, that relationship does exist. But for right now, this change in suitability within forested areas of the Tongass may not indicate much about changing cover of salmonberry in that particular region. So with all of this in mind, potential threats to occupancy, changes in cover, it might call to mind, and what I thought was the next logical step was to say, well, what environmental attributes are associated with greater cover of these species? Are there attributes of the forest that managers could be focusing upon that maybe could help them to manage for increasing access, knowing where to build trails for folks to access these resources? Or if you're planning forest management activities and preserving these species as your goal, what could you be aiming at? So to get at this question, I used FIA data again, but also worked with the Alaskan Youth Stewards Program, which is a great program. It takes high school age interns out into the forest to do forest management and science activities. And the data that they collected served as a validation data set for a generalized additive model that I built using FIA data. Lots of variables, kind of spaghetti at the wall, were thrown into this generalized additive model, and then model selection kind of helped to identify what are the important predictors for predicting the cover of blueberries and salmonberries on these plots. And I'm just going to be showing you some of the results of this over here on the left. This is for the blueberry model. The generalized additive model significantly outperformed a null model. So it's a fairly good fitting model. And if we look at it compared against withheld data set of FIA data, significant, strong, positive relationship, it does a pretty good job. And the same is true when we look at it against this Alaskan Youth Stewards data. When we look at the predictors that were retained in the final model, shrub cover comes out as being far and away the most important, but this isn't super informative because blueberry is the most common understory shrub in the Tongass National Forest. So of course, that's going to be an important predictor for cover of blueberry is how much blueberries are basically. But if we look at the other stand level predictors, stand size, age, and forest type shook out as being pretty important. And if we look at these sort of response plots here, blueberry cover tended to be highest in stands with smaller trees that are less than 50 years old, and in hemlock and yellow cedar stands. So that might give managers an idea of where they could be aiming either management activities or increased efforts towards, increasing access to these shrubs. Salmonberry model wasn't as great. This seems to be a theme, but it did still significantly outperform the Knoll model. And there was a significant positive relationship between model predictions for salmonberry cover and actual cover recorded on FIA plots within this withheld data set. No such relationship with the data collected by the youth stewards. So all this indicates is that there are probably variables that could be included in these models to improve them. My guess is probably something to do with disturbance, as salmonberry tends to be associated with disturbance, but that wasn't included in the model, so that may account for some of the reason why this isn't a great fit. Far and away, again, forest type is a really important predictor for salmonberry cover, and if we look at that relationship, it tends to be highest in hemlock and Sitka spruce stands. Sitka spruce makes a lot of sense because it also tends to be associated with disturbance. So that kind of passes the dummy check there. So that's a lot to spit at you. Just very quickly, some of the take-home messages is that occupancy is expected to remain high, which is great, but cover may decline dramatically for blueberries, not so much for salmonberries. So that is concerning, depending upon obviously if that relationship is conserved. People do not care necessarily about cover. What they care about is fruit availability. So that, I think, is the next really necessary step in this research, is connecting those two and seeing where does one translate into the other. And then I think identifying these environmental attributes that are associated with higher cover could potentially give managers a little bit of an idea of where they could start if their goal is to ensure continued access to these subsistence species that are really, really important from a variety of perspectives. So with that, I just want to thank my funders on this, the Research and Underserved Communities Fund, all of the FIA crews and data managers that make these data available, and my collaborators on this project, Di Johnson and the Sitka Conservation Society, and definitely the Alaskan Youth Stewards Program, and all of the tribal and community organizations that participate in that program. And I probably have gone over time, but if I haven't, I'm happy to answer questions either here or in the chat.
Awesome. Thanks, Katie. Yeah, really interesting work and certainly really tangible for communities here in Alaska. We are at the end of our session time here. So I think if anybody wants to ask a question, they can until we get the boot. Otherwise, you could drop it in the chat or reach out to Katie or any of our speakers. But before we do that, I just want to say thank you to our speakers for a really great session. It was, yeah, we ran the gamut from understory veg to nationwide sulfur and nitrogen deposition. So really appreciate the breadth and the depth of our talk today, of our sessions today. Yeah, I guess we'll just leave it at that. Thanks again, audience, and we'll see you next time. Thanks. Oh, we have a couple. I guess Mark Ambrose has a question.
Yeah, just a thought, Katie. I was wondering, did you try, since you found that shrub cover was a big predictor of blueberry shrub cover, did you try just pulling that variable out of the model and then refitting everything else? Because
I did.
Yeah, you did?
Mm-hmm.
Okay. Because sometimes when you pull it out, the other things will pop out.
Things get, they did for sure. And I thought worth including because, there's an obvious relationship there. And I felt like, yes, that's one to explore and explain, but pulling it out definitely, I don't think it shook up the model terms that were included too much. Their rankings might have changed a bit, but I think it was still the same predictors that were retained, but the model fit definitely went down, which makes sense when you take out a predictor that's basically not a one-to-one predictor, but pretty close of your response. But I believe it's been a while since I've looked at it, but I believe they were like a similar set of predictors.
Right. Thanks, Katie. Thanks.
Thank you. Thanks, Mark,
Thanks,
for
everybody.
that question.
All right, we'll have to leave it there. Thanks, everybody.